Okay, so here we have some Lego bricks, and these ones here on the left represent a DNA sequence that we've amplified. And basically in a Sanger reaction, these individual bricks here represent DNTPs, and these ends without studs on them represent DDNTPs. And we know that the DNTPs contain the 3' hydroxyl group, which allows for extension of further bases after each consecutive base is added on. Whereas the DDNTPs don't contain that hydroxyl group at the 3' end of the molecule, and so therefore no further DNA can be extended off that end, therefore basically ceasing the extension or the continuation of the DNA sequence. Now in our sequencing reaction, we essentially have DNTPs and DDNTPs, which are made up of these kind of blocks here that you can see. And they're added into the sequencing reaction basically at random by Taq polymerase as it's extending off the DNA molecule. And so with every additional base that's added, either a DNTP is added, allowing for the continuation of sequencing to follow, or a DDNTP is added, stopping any further extension of the PCR product. So because Taq polymerase basically has this mixture of DNTPs and DDNTPs to be assembling the new DNA strand from, it's randomly pulling these pieces out, essentially the same as what you're seeing here with the Lego pieces, just pulling them out at random and getting either a DNTP or a DDNTP. And if it gets a DNTP, continues adding on more and more nucleotides to the newly generated DNA strand. And if it gets a DDNTP, without essentially the studs like on a Lego block, you just can't build any further on from that. And so you see here, as we're going through, we're generating all of these new fragments of DNA at various lengths. Now, remembering that in Sanger sequencing, those DDNTPs are also tagged with a fluorescent dye. So what that means is that end block is fluorescently tagged with a dye that reflects the nucleotide that it is. So in this example using the Lego, we could say that the blue blocks are a blue dye, that would be say a FAM dye, that's representing an A molecule. And the yellow ones might be a T, and the red might be a G, and the green might be a C, for instance. And so those fluorescent molecules on the end of each of the blocks, when they go through the capillary, they all move at different rates because they're different sizes. And then the fluorescence is detected on the basis of the length of that molecule. So you can imagine this continuing on and on and on, and generating lots and lots and lots of extra copies of various length molecules, all possessing a fluorescent tag at that last position. And the end result, after millions and millions of tiny reactions, cycle after cycle of the PCR, is that we end up with all of these different length blocks of all of these different length DNA molecules, all possessing that fluorescent tag at the final 3' position. So now you can imagine all of these blocks being run through a capillary on a capillary electrophoresis instrument. They're all going to migrate at different rates, with the smaller blocks moving faster through the capillary than the larger blocks. And so the smaller blocks pass through the sensor or pass through the detector within the capillary, and that fluorophore on the final block is excited. And so the one-piece block, we get a red fluorescence. The two-piece block, we get a green fluorescence, then a blue, then a yellow, and that is how we're able to tell the sequence of the DNA with Sanger sequencing.